Okay, let's get into it. The compound microscope. I know, that thing can look pretty intimidating sitting on the lab bench. But trust me, by the end of this, you're not just going to understand it, you're going to feel like you've mastered it.
And let's be real, that pre-lab quiz is probably on your mind. The practical feels like a huge deal, and you want to get it right. Well, don't you worry. We're going to walk through every little thing step by step so you can walk into that lab feeling totally confident.
So, here's our roadmap. First, we'll get clear on what your mission is for Lab 5. Then, we're going to get properly introduced to your microscope, breaking it down into simple parts. After that, we'll learn how to get a perfect view every time. We'll observe the key differences between plant and animal cells, and then we'll wrap it all up by making sure you can ace that pre-lab quiz.
All right. First things first, your mission. Look, before we even touch a knob or a slide, we got to understand why this lab is so important. This isn't just about looking at tiny stuff, it's about seeing the actual architecture of life itself.
And the whole reason this matters comes down to something called cell theory. This is one of the biggest ideas in all of biology. It basically just says that everything that's alive, from a tiny piece of algae to you, is made of cells. And the cell is the smallest possible thing that can be considered alive.
So what skills are you going to walk away with? By the time you're done, you'll know how to handle the microscope like a pro, you'll be able to name all its parts, you'll get the key vocab down, you'll prepare your very own slide from scratch, and you'll be able to spot the major differences between plant and animal cells. Easy enough, right?
Okay, let's get to the main event, the microscope itself. Yeah, it looks complicated with all the lenses and knobs, but I want you to think of it as a key that unlocks a hidden universe you can't see with your own eyes. We're just going to break it down into a few simple systems.
And before we do anything else, here is the #1 most important rule. Seriously, you always carry the microscope with 2 hands. One hand grabbing the arm, nice and firm, and the other hand underneath supporting that heavy base. It protects the instrument, and honestly, it immediately shows you, instructor, that you know what you're doing.
So how do we get those crazy magnification numbers? It's actually really simple math. The eyepiece you look through, that's the ocular lens, it always magnifies things by 10 times. Then you just multiply that by whichever objective lens you're using. So the little 4x objective, that gives you a total of 40x magnification. The 10x gives you 100x, and the 40x gives you a whopping 400x. That's all there is to it.
Now to make remembering all those parts a lot easier, let's just group them by what they do. You've got the magnification system, that's the lenses that make things bigger. The illumination system, all the parts that control the light. The focusing system, those are the knobs that make the image sharp. And finally, the support system, that's just the frame holding it all together. See? So much easier to remember that way.
Okay, we know the parts, we know the theory. Now it's time for the fun part, the hands-on skills. This section is all about the how-tos, the exact steps you need to follow to get that perfectly crisp, focused image every single time.
So this 6-step process is everything. #1, you always start with the lowest power. That's the 4x objective. 2, center your specimen right over the little circle of light. 3, and this is super important, watch from the side of the microscope as you use the big, coarse knob to raise the stage all the way up till it stops. 4, now you can look through the eyepieces and slowly turn that same coarse knob the other way to lower the stage until you see something. 5, use the little, fine knob to get it perfectly sharp. And 6, always recenter your image before switching to a higher power.
Now, when you start moving the slide around, you're going to notice something kind of trippy. When you physically push the slide to the left, the image you're looking at actually moves to the right. When you push the slide away from you, the image moves toward you. Everything is completely flipped and reversed. That's just how these compound microscopes work.
Now here's a word you absolutely have to know for your quiz, parfocal. This is an awesome feature on modern microscopes. All it means is that once you get a perfect focus on that lowest power, the image is going to stay almost perfectly in focus when you switch to the 10x or 40x. You'll only need a tiny little turn of the fine focus knob to sharpen it up. It's a huge time saver.
And its partner is another key term, paracentric. This just means that whatever you put in the dead center of your view will stay in the center when you switch to a higher power. And that is exactly why step 6, centering the image, is so critical. If you don't center it on low power, when you zoom in, it'll be way off to the side, completely out of your view.
You've mastered the tool. Now it's time to be a scientist. We're getting into the real hands-on investigation, where you're going to prepare your own slides and see these building blocks of life for yourself. This is where we stop just learning how to use the equipment and start using it to answer a real scientific question.
So you'll be making what's called a wet mount. It's a fundamental lab skill. Step 1, a single drop of water in the middle of a clean slide. Step 2, put your specimen, like a tiny leaf or some of your own cheek cells, right into that water drop. Now here is the pro move. Take the little plastic cover slip, touch one edge to the water first, and then gently lower it down at an angle. This is the trick to avoid trapping those super annoying air bubbles that can totally ruin your view.
So, what are you actually looking for? This isn't just a scavenger hunt. You're running a real experiment. The thing you're changing on purpose, the type of cell you're looking at, either plant or animal, that's your independent variable. The things you observe and measure, like whether there's a cell wall or its overall shape, those are your dependent variables. They depend on which cell type you're looking at, right? And to make it a fair comparison, your constants have to stay the same, like using the same magnification and the same slide-making technique for both. This whole structure is what turns just looking at stuff into actual science.
All right, we're in the home stretch. You've got the theory, you've got the procedure, you even have the experimental design down. Let's lock in these last few concepts so you can walk into that pre-lab feeling like you own it.
Here's a classic quiz question that really gets to the heart of how these things work. Just think about it for a second. Why does the sample have to be so incredibly thin, almost transparent?
The answer is literally in the name. It's a compound light microscope. The light at the bottom has to shine up and pass through your specimen to get to your eye. If your sample is too thick, it just blocks the light. You won't see anything but darkness. The light has to be able to get through. Simple as that.
Okay, one more. This is a huge one, both for safety and for your quiz. Why is it an absolutely terrible idea to even touch that big coarse adjustment knob when you're using the high-power 40x objective?
The answer is a term called working distance. That's the tiny bit of space between the tip of the lens and your slide. On high power, that gap is paper thin. The coarse knob moves the stage a lot with just a small turn, so if you use it, you will literally crash that expensive lens right into the glass slide. And trust me, that's a very bad sound. Always only use the fine focus knob on high power.
Okay, let's wrap it all up. This is your ultimate cheat sheet. Total magnification is just the ocular lens times the objective lens. Always start focusing on the lowest power, 4x. Remember, the image is flipped upside down and backwards. Center your object before you zoom in because the scope is paracentric. And I'll say it one last time, only use the fine focus knob on high power. And for your investigation, the big takeaway, plant cells have a rigid cell wall, a perfect example of a dependent variable you'll be looking for.
And I'll leave you with this one last thought to take into your lab. As you're looking at these cells, seeing how a plant cell's rigid wall gives it structure while an animal cell's squishy membrane allows it to move, ask yourself, how does that one tiny difference at the cellular level completely change how these two organisms are built to live and survive in the world? That's how you connect what you see in the microscope to the biggest questions in all of biology. You got this. Good luck.